Okay, so hello everyone, hello. So today this is another class. So we'll be doing the today we're gonna be doing the plasma membrane. So we're gonna learn about what the heck was this. Wait, no, wait. Yes. We're going to learn about the plasma membrane. So I hope you could hear me. No. What is the plasma membrane? Now, if I have the shell, so I'll draw a shell here. Okay. Alright. Okay, have this shell and all that. I'm not gonna go much into detail, but then this is what our cell looks like with more parts and all. I'll label them later. Now you notice that there is this membrane. Okay, this membrane, which is called as the plasma membrane. Which is called as the plasma membrane. Now, this plasma membrane is semi-permeable for solvents, and it is selectively permeable for solutes. Now, if you look at this membrane, it's made up of something. And it's not empty like this. Okay. So it's made up of proteins. Wait, I started writing too big. Oh no, shit. So it's made up of proteins. Dude, what happened to my mark? Okay, sorry, my bad. Proteins and lipids. Fine, it's made up of proteins and lipids. Now, this is the cell membrane or the plasma membrane. You also call it the. They also have a fancy name, also called as the plasma, which we really don't need to focus on. Fine, so don't focus on this at all. Prepare. Fine, okay, now if you look at this, you saw that this is semi-permeable. So let's say we have an object that could basically go through this if it's the molecule of a solute like H2O. Okay, now if it was a solvent, then it would have to be allowed or given a pass by the by the cell membrane so that it could pass through. Fine. Now there are several ways by which this molecule can go inside the cell. Now let's say you have more molecules of water, okay, H2O, okay, and here you have one molecule of H2O. Now, so all these molecules go here. So all these H2O molecules go here. Basically, it means that it goes from high concentration to low concentration. Fine. Now this can take place through two methods. Movement can take place through two methods. They are osmosis and diffusion. Osmosis and diffusion. Now, what is the major difference between these two, which we will discuss later? Now, what is osmosis and what is diffusion? First, let's understand diffusion. Now, diffusion, like molecules of any medium move from a region of high concentration to a region of low concentration. Fine, got it? So this is the definition for diffusion. So diffusion is like any medium can go through. Now, basically diffusion does not have a semi-permeable membrane. Now let us look at osmosis. Fine, now what is osmosis? Here that we have a semi-permeable membrane exists, has a semi-permeable membrane. Fine, so that is the difference. And also here osmosis takes place in liquid only. Now in this osmosis, we have two types. Okay. Endosmosis and exosmosis. Now, what is the difference? Basically, endosmosis means that a substance, right? A substance is going inside the cell. So, like, for example, we have a large number of H2O outside the cell. It's going inside the cell. Fine. Now, this one means that something is going outside the cell. Now, if you look at the structure of the plasma membrane, like there were two scientists called Singer and Nicholson in 1972. They proposed the plasma membrane layer, which is known as the fluid model. Okay, fine. Fluid mosaic model. Fine. Now, so here it suggested that the plasma membrane is made up of a bilayer of phospholipids. Bilayer of phospholipids. Now, okay. So basically, it consists of two types of proteins: intrinsic and extrinsic. Wait. Okay, fine. I think it's intrinsic, yeah. Okay, so this protein basically spans the entire bilayer. Okay. This protein basically spans the entire bilayer. On the outer membrane of the phospholipid bilayer. Okay, so we'll end the session here because I'm in my hometown and making this. So this is all that we have done about this chapter. What I'm doing is that like you get more into detail. It also covers a few chapters, few concepts from 11th. Okay, so bye guys.